Today, I'm going to talk to you a little bit. I'm going to give you a little primer on vaccines, a little science lesson behind how they work, what kind of vaccines we have out there. And then I'm going to give you a little bit of story about our own vaccine and where it's at right now. So just to start out with the vaccine story I'm going to tell you about, it's a biotech company here in Seattle called HDT Bio has picked it up to commercialize it and advance it to clinical trials. And I have no financial interest in this company. And so here we are on actually August 5th, 2020. And, we're at over 18 million total confirmed cases, over 150,000 deaths in the United States. And the only ways right now we really have of controlling this pandemic is through social distancing, and to slow the transmission, and testing to identify cases and quarantine. Ultimately, the way to stop a pandemic is through vaccination, by inducing immunity in the population to be able to slow and eventually stop the spread of the virus. And so what are vaccines? Just to kind of start out with, vaccines are really just a way of priming the immune system. We call that immunization, to provide protection from the disease that's caused by that pathogen without actually subjecting that person to the disease itself. And so, whoops, I'm losing control of my panel here. So, we think of vaccines as an ounce of prevention, a pound of cure. And, here you can see, back in the 60s, we developed there was a measles vaccine that was developed, and prior to that vaccine, you can see the incidence of that infection over time. And upon licensure of that vaccine, you can see how very quickly that pandemic or that epidemic was eliminated. And this wasn't accomplished necessarily by immunizing every person. What vaccination achieves is something called herd immunity. And what occurs there is that, right now we're in our pandemic where we have no one immunized. And so you have a lot of the spread of the virus and sickness and illness. Once the virus starts to spread, you do start to get people to build up immunity in the population, but it's not enough. The virus is still able to find people and infect it. It's sort of an equal opportunity invader. It pretty much invades anybody it can get to. But ultimately, what we try to do with vaccines is something that we call herd immunity, which means that you're going to get enough people vaccinated in the population, not necessarily have to vaccinate everybody, but enough so that the virus runs out of places it can go. And so once it does that, then you start to see this immediate drop in terms of the infection rate. And that's really ultimately the goal of what we want to do with the vaccine for this particular pandemic. And so immunization actually can occur in two different ways. What we're going to talk about in terms of vaccine is what we call active immunization. And that's really where you're going to use either a vaccine or even natural infection can be theoretically called active immunization to stimulate an immune response. It's going to provide you protection from the virus. An important thing about active immunization is specific, which means that when you get vaccinated against a virus, that vaccine is going to protect against that virus, not other ones. And it has memory, which means that if, a year from now, this same virus emerges, your immune system remembers it and is able to respond to it quickly and provide protection. Another type of immunization that you're hearing about is called passive immunization. And that's when you take plasma, convalescent plasma from patients who just recovered, and you can transfer that to other people and provide them with short-term immunity. You're basically borrowing antibodies from somebody else and transferring it to another person. What's also in development right now is something called monoclonal antibodies, and that's where they've basically take some cells from a convalescent patient and engineer them to actually now produce some antibodies, a single type of antibody that is very potent against the virus. And they can manufacture that at high amounts and use that for passive immunization. The important thing about that is that's just ready-made immunity. It's going to give you transient protection, but it has no memory. So as soon as that antibody declines, that individual, again, will be susceptible to the disease. So what we're trying to achieve, of course, is active immunity. And so when we think about vaccines inducing memory, there's really two different stages to that. When you're going to hear that most of the vaccines that are being developed for coronavirus are going to take a priming immunization and a booster dose. And what the priming immunization does is it's kind of get your immune system ready. You can get some antibody titers that come up, and sometimes this level is enough to give you protection transiently, but it quickly wanes. Now here in this particular gap, what's happening is that your immune system is making that memory. It's remembering now that first exposure. And then when you get a second exposure to the virus, then all of a sudden those memory cells are going to expand and they're going to make a much quicker response to the antigen, and the immune response is going to last longer as well. And so, so this is really kind of what we want to achieve, because this longer lasting immunity is what we're going to need, at least 6 months, at least to a year immunity, if not a lifetime immunity against the virus. Sometimes you can get certain types of vaccines that will be able to induce this primary response and still induce an increasing response without requiring the booster. We'll talk about that in a minute. And so I'm going to give you a quick immunology lesson, because this is really important for vaccines for coronavirus, and that is how are these vaccines actually inducing an antibody response. And so when you get a vaccine, you're getting basically a piece or a component of the pathogen, or it might be the whole pathogen that's been killed. And you have cells in your body, and they're called antigen presenting cells. And they basically pick up that vaccine, and now they're going to take it to your immune system and they're going to present it to it. And vaccines are often formulated with something we call adjuvants. And adjuvants are just basically, they're helpers. They help the vaccine get seen better by your immune system, and they act on something that we call T helper cells. So this antigen presenting cell is going to talk to this T helper cell, and they're going to get together, and they're going to go talk to what we call this B cell. And this B cell is what produces your antibodies. When it gets a signal from the T helper cell along with the antigen presenting cell, it's getting told to make antibodies that are specific to the coronavirus. And so once these plasma cells are made, they're presumably long lasting, and they can provide immunity for many years after a vaccination. And so, you know, how ultimately is a vaccine for coronavirus going to protect us? How are these antibody responses that we're going to induce against SARS-CoV-2, the virus that causes COVID-19? And so let's just take a look at the virus. You've all seen this picture from the CDC, and it's decorated with these little red things, which we call the spike protein. Well, this spike protein is actually how the virus gets into the cell. It uses these little red things to attach to the cell, and then it's able to get in there. And so what we're trying to do is prevent the virus, block the ability of the spike protein to attach to the cell and get in. And so within this particular spike protein, there's a little region, it's called the receptor binding domain, or RBD. And there's a receptor on our cells, and it's called the ACE2, okay? And we all have them. And this receptor binding domain on the spike protein is what attaches to our cells and actually gets inside. So what we're trying to do with a vaccine is we're going to make these antibodies look like these Y shapes here. And if these antibodies attach to the receptor binding domain, or the spike protein, that's going to prevent the virus from being able to get into our cells. And so that's really in a nutshell how you know, a vaccine is going to work. I kind of covered a little science here. Are there any questions right now that were not clear for you that I can answer? Now I kind of want to move forward just to kind of talk to you about the different types of vaccines that are under development for COVID-19. What we're used to right now are just these traditional licensed vaccines. Most of our vaccines fall into one of these 4 categories. We have live attenuated vaccines, and that's things like our measles vaccine. And basically what that is they took the virus and they put some mutations in it so that it can replicate a couple times in your body, but not enough to cause disease. Once your body sees that live attenuated virus, it then makes an immune response, and that immune response can protect you from the real wild type virus. We also have inactivated vaccines, and that's basically where you just take the virus and you kill it, and then you just inoculate that. So we have every year an influenza shot, and that actually is an inactivated type of vaccine. Another type is called a subunit, and that's just basically where we take a part of the virus, like the spike protein, for example, not the whole virus, and it's basically produced in the lab in a dish, and then it's purified, and then it's injected into people. And then you make immune responses only against that part of the virus. Our hepatitis B vaccine is a good example of that. And our final category is what we call a toxoid type of vaccine. And that's really just where you take a particular, it's basically like a subunit, but it's the toxic portion of the virus. And we produce that in a dish, and then we inject that, and we make antibodies against that particular toxoid. And so those are the sort of what we coming into COVID-19 we had in our toolbox in terms of licensed vaccines. So what are we doing now? We have all kinds of new sort of approaches. We do have
Dr. Fuller?
Yeah, go ahead.
If I could stop you for a second, I do notice that 3 hands went up when you asked a second ago, and it took me a second to notice that. So let's go grab those 3 questions. We'll go to Wait first, Wait, and then Adriana, and then Kira, and then we'll go back to the presentation.
Sure.
Thank you. Just a quick question on the science, 2 little questions. The first is, you've been focusing on antibodies, but so it begs the question of whether the antibody response we know to be sufficient to induce immunity to this particular disease, or whether other components of immunity, cellular immunity or innate immunity, are also necessary. And second, have we determined whether the APC cells are presenting the specific epitopes that we are making vaccines to replicate?
Yeah, great questions. And I am totally going to get to the T-cell response, because I think that the T-cell responses are going to be important. In a nutshell, if you have really good antibody titers, if you develop them to sufficient levels, antibody is usually sufficient to block the infection. But not everybody is going to develop the same levels of antibody. For example, the elderly might not develop as good antibody, or immune compromise. And so this is, we have a second layer of defenses called a T-cell response. And if the virus gets past the antibody, then the T-cell responses can kick in. So I believe that T-cell responses are going to play an important role, as well as antibody, in protecting against this particular virus. And then your second question was related to antigen presenting cell, and whether we knew whether or not it was actually presenting the correct epitopes. We do know that, just basically by mapping how the immune response responds to both the infection as well as the vaccines, we can actually show that the antibody are directly targeting the receptor binding domain of the spike protein. And that means that the antigen presenting cell is appropriately presenting that particular epitope. Great questions. What else?
Adriana?
Yeah, I think you might be getting to my question in literally the next couple minutes now that I'm looking at your graphics. But I wanted to ask about the difference between the RNA vaccine and then the protein vaccine that we had recently just saw in headlines with Novavax. But since you're getting into that, the one thing I would like to know after that would be, why are experts saying that the protein vaccine might be better than the RNA one?
Yeah, those are great questions too. I think I think it's still out there as to whether protein vaccines versus nucleic acid, which includes RNA and DNA vaccines, would be better. But we know from many years of vaccine development, like the hepatitis B vaccine and other sorts of vaccines, that recombinant protein vaccines, these subunit ones is what they're referring to, are very, very effective. One of the limits of subunit vaccines is they take a very long time to develop, because you have to manufacture them in cells, you have to purify them. They have a much longer process. And so DNA and RNA are much quicker. And I think that both of these are going to play an important role in terms of protecting the population. I don't think it's going to be so much a competition, but a partnership between these types of vaccines.
Okay, and Kira.
Hi, thank you. I just had a question on the underlying technologies involved. Does the different approach affect the length of immunity that each vaccine is expected to confer?
I'm sorry, I sort of missed that.
Basically, depending on the technology used to develop the vaccine, is there expected to be different responses in terms of immunity, in terms of how long immunity lasts, depending on the technology used?
Yeah, and that's exactly what I'm going to get into. Great primer for my next conversation about these different types of vaccines. They make a huge difference, not only with respect to, say, how durable they might be, but also with respect to what types of immune responses that you're going to induce. So when we talk about the whole inactivated, and that there are a couple companies in China that are now actually very far along in developing an inactivated. That's where they just kill the vaccine. That particular type of vaccine is very effective in inducing antibody, okay? But when we talk about these new kids on the block right here, and that's this recombinant viral vector that you're hearing about, that would be from the Jenner Institute, for example, with AstraZeneca. It's making a chimp adenovirus-based viral vector. That's basically what you do is you use an adenovirus that's like a cold virus, and you manipulate its genetic material. So you take out the genetic material that is part of the adenovirus and replace it with a gene expressing the spike protein. And now you've got this viral vector that is, virus which is harmless to us, it doesn't really cause us any disease, being able to get inside your cells and deliver genetic material and instructing your cells to make spike protein against the coronavirus. Now, this type of vaccine, because it can get inside our cells and present to antigen-presenting cells that way, is able to stimulate not just antibody, but also very robust T-cell responses. And we just talked about how both those types of responses could be important. RNA and DNA vaccines are the same, in many ways, the same as kind of recombinant viral vectors. The difference is that they don't have a virus carrying them in. Instead, it's just DNA or RNA getting delivered directly into our cells and then instructing our cells to start making spike protein. And we'll get into a little bit more difference between these two. One of the problems that you have with recombinant viral vectors is that you start to build immunity against that virus that's carrying that genetic material. You start to make antibodies against the adenovirus itself, okay? Once you start doing that, the next time you want to come back in and boost, remember we talked about boosting response, now it's not going to be as effective because now your body is fighting against the vaccine itself. One of the advantages of DNA and RNA is that you get them in, they don't have that virus around them. And so you're not going to have this sort of pre-existing immunity getting built up against the vaccine itself. So you can keep boosting with that. Say if the antibody responses are starting to wane and you need to boost those back up, you're going to have a lot easier time with DNA and RNA than you would with a recombinant viral vector. So that's sort of kind of in a nutshell, those different types. There's a recombinant subunit protein that this is being developed. This is recombinant viral vectors. And as I mentioned, the Ho inactivated are really the main types. And we'll get into that a little bit more in the end when I start talking about where we're at with clinical trials and the like. But you can see that there's a lot of different vaccines in development. And this is actually an old figure now. It's several months old. But as you progress a vaccine from the exploratory phase into clinical trials, it becomes fewer and fewer. And that's just because, we test these out and we end up, throwing some out and advancing other ones forward based on, how they perform in preclinical studies. We first started developing a vaccine for SARS-CoV-2. We thought about really what are the key attributes that you're going to need for an effective pandemic vaccine. And there are really 4 main categories. And I think, first of all, it's going to need to induce immunity quickly. If you have to have 4, 5, 6 doses to get to an immune response that's protective, that's going to take a long time to build that immunity in the population. So ideally, it'd be great if we could get that immunity within a single dose. But as I mentioned, 2 doses might be required to achieve that, and that's still okay. We talked already about antibody and T-cell responses. I think it's going to be very important to not just induce antibody, but also T-cell responses. And I'll talk to you a little bit more about what these cytotoxic T-cells are in a minute. Also, it's really going to need to be effective across different demographics. So as many of you may know, vaccines don't work as well in the elderly or the immune compromised or certain people with comorbidities. And so, especially because a lot of these are the most vulnerable to SARS-CoV-2, we're going to have to make sure these vaccines are going to be effective across all demographics, not just the young and healthy. And finally, I think really important is that it needs to be fast and cost effective to scale up. Ideally stable at room temperature. And, you know, that's really important because, you know, once you have a vaccine that you've demonstrated works in clinical trials, you're going to want to have that scaled up really quick to billions of doses to get that out very quickly. And you think about how most of our vaccines are done, we're giving them at you know, childhood, and each vaccine companies might be producing several hundred millions of doses of vaccine a year just to vaccinate the population in a rolling sort of cycle year by year. In a pandemic, you need to suddenly have billions of doses very, very quickly to vaccinate the population. The other thing that is going to be real important, I think, is that it's stable. Most vaccines require what we call cold chain, which means they have to be in the refrigerator or freezer to distribute. And that's going to cause problems with actually getting distributed worldwide really quickly. So if you can make a vaccine that's stable at room temperature, that's going to be even better. We talked a little bit, you know, about where our area of research is really in what we call nucleic acid vaccines. These are DNA and RNA vaccines. I say they're new kids on the block in quote marks because they're new to, you know, I think to the general public, but certainly for our scientists, we've actually been working on these types of vaccines for over 30 years now. And, you know, as I explained in, you know, already, what these two types of vaccines hold in common is that they are basically codes for the particular protein from the virus. In this case, it would be spike protein that get injected into our own cells and then instruct our cells to start making the vaccine and then stimulate the immune response. The difference between DNA vaccines and RNA vaccines is significant. DNA vaccines, they have to get inside your nucleus, okay? And then they transcribe into RNA and then they produce protein. Okay, big advantage of DNA vaccines is they're much more stable than RNA vaccines. But the disadvantage is that they need special delivery techniques to be able to get them inside the nucleus. The RNA vaccine has the advantage there in that it doesn't need to actually go inside the nucleus. It just needs to get inside your cytoplasm. And that's easier to do, so that every cell that gets the RNA is going to actually be able to produce the protein. So generally, if you get a successful RNA vaccine, it's going to produce more protein generally than a DNA vaccine, and more protein usually means a higher immune response. Any questions about the difference between DNA and RNA? So we have two companies, basically Inovio is a company that's been developing a DNA vaccine, but there's a number of other ones that are also developing a DNA vaccine. Of course, you're familiar that there's Pfizer and BioNTech and Moderna and J&J and a bunch of other groups that are actually developing RNA-based vaccines.
Okay, we do have a couple questions.
Great.
We'll go to Taryn and then Matthew and then Umair. And I guess I'm watching the clock, so try to keep the questions fairly tight so we can make sure we get through as much of her presentation as possible. We'll go to Taryn first.
Hi, thank you. Yeah, I'm Taryn. I'm a reporter out in San Diego. You just mentioned we've been working on DNA, RNA vaccines for about 30 years, but this is the most I think any of us have probably ever heard of these. So what's been the challenge? I mean, is there a common challenge in the past that's prevented these vaccines from becoming licensed? And is there something different about the COVID pandemic that means that these are more promising?
Yeah, great question. It hasn't been. They've just been going through the usual process that, you know, when vaccine discovery, there usually is a pretty long timeline from the time that we first figured out how that you could take a DNA, basically a naked DNA, and inject that into people and make your own cells produce your vaccine. What happened in the early stages of DNA and RNA vaccine discovery is that the first vaccines we tested looked great in animal models, were great in preclinical studies, but the first DNA vaccines, and then DNA was the first one to get in clinical trials, they were utter failures. This was in the early 90s. And so everybody was into DNA and RNA vaccines at the time, and when everything failed, everybody and their grandmothers got out of it. So except for a few of us, we hung in there and we said, well, let's solve this problem. And so since the 90s, there's been a lot of effort to actually, and I won't go into all the different technology improvements that were done, but they basically, a number of improvements revolutionized both DNA and RNA vaccines that now we can actually see that they are working very well in people. And so COVID-19 came, and DNA and RNA vaccines were already in phase 1 and 2 clinical trials for other infectious diseases. And so it was a very quick pivot for them to swap out that genetic material for the spike protein and rapidly move forward into clinical trials. So that's sort of the story behind them.
Okay, Matthew.
I'm Matt. I'm a freelance science writer. You mentioned that you'd have to inject these into the cells, and I guess for DNA into the nucleus. Is there a way to inject them into the blood and have them get their way into the cells?
That's not as efficient. So usually DNA and RNA vaccines are best, more sufficient. They can access more cells and actually get into the cells better by injecting them either into the muscle or into the skin. And there, it's just the reason is that you just have more compact and cells are more available to inject. But with that said, people have injected them into the blood and intravenously, and you can certainly get DNA and RNA vaccines in the cells. It's just not as efficient, number one, and number two, for vaccines, intravenous delivery of vaccines would never be really practical. So
you have to stick the needle into like a bunch of cells in a person?
Yeah, just a muscle, just like you get your
or
regular.
Just one cell?
Yeah, you just inject in the muscle. And then with DNA vaccines, there's a technology called electroporation. It's basically not just a needle goes into your muscle, but there's a little electrical pulse that opens up the cells so that the DNA can go inside that. Does it hurt? Yes. As much as the needle hurts, yeah, that little electrical impulse there, that does hurt. That's a company called Inovio that's developing that. So there's another way of delivering a DNA vaccine that actually was developed in my lab, and it is where you actually use what we call gene gun to accelerate tiny little microparticles into the epidermis of your skin cells, and then those skin cells start to produce the vaccine, and that doesn't hurt. So we're sort of more of the earlier stages in developing that technology, but that's another way of delivering DNA vaccines. RNA vaccines, I'm going to get into how those are delivered in a second here.
Okay, and final question this round from Umair.
Hi, Umair Irfan at Vox. Just based on your last comment, I think I'll just wait until you respond and get to that.
Oh, okay, great. All right, so let's talk about the RNA vaccine that we developed. I'm going to give you a quick story about where we're at with that. And so we developed a novel RNA vaccine, and what happened was in January, the genome sequence first became available for SARS-CoV-2, and a postdoc in my lab called me up late at night and says, can't we make this vaccine? And, totally, as I mentioned, with RNA and DNA vaccines, you really only need actually the genetic sequence from the pathogen to start to make the vaccine. With other types of vaccines, you need the pathogen itself. So that's why DNA and RNA vaccines got to take off really quickly. Within a week, it takes very quick to actually design one. We already had pilot DNA and RNA lots to start testing. And then by February, we were already testing them in animals, and we were testing various different constructs, as we call them, to see which one might work better initially in animals. And now today we're, in August, the company I told you about is now advancing that to clinical trials, or they're manufacturing it and testing it for toxicity and the like. And we're anticipating this vaccine is going to go into clinical trials at the end of August and the beginning of September. So the main thing is that within only 7 days after an outbreak, or the sequence for an outbreak has been identified, we could have already vaccines being tested and evaluated. So that's how fast, and only DNA and RNA vaccines can really work that quickly. And so, we think of nucleic acid vaccines as really one of the best technologies out there to develop, to work against pandemics. And so, as I mentioned to you before, that DNA vaccines, they require that nuclear delivery, right? Whereas RNA vaccines just require to get only into the cytoplasm of the cell. But basically, they work both the same. The DNA transcribes into an RNA, and then once you get RNA, it makes protein vaccines. Now, they're very different from traditional vaccines because you're going to require all that cell-based manufacturing process and purification before they get injected. Whereas here, you've got the DNA and RNA ready to go already into people. Whereas now, a lot of these traditional vaccines, these protein vaccines you're hearing about, they're going to be later to the finish line. Although what they're trying to say is that we're going to be better. When we get there, that remains to be seen. For sure, DNA and RNA are the fastest, and they can be rapidly scaled up, as I mentioned, and they can be stable at room temperature. We talked about why that would be really important. And finally, one of the things that's really important, I think, about DNA and RNA is that they're capable of inducing both those antibody and T-cell responses, which are really two weapons of defense. And I'll talk about that in a little bit. But I want to tell you a little bit about our vaccine. It's an RNA vaccine as well. And of course, there's Moderna and there's Pfizer developing an RNA vaccine, but how is ours different? Well, what happens is with ours, we have, basically we start with the same thing. We start with the DNA and then we make the RNA vaccine. And what we do is we actually include in our RNA vaccine another code that instructs that RNA to start replicating itself. So we make more and more copies of that RNA. And once we make more copies of that RNA, it makes more protein, more vaccine protein. And more protein makes a stronger immune response. And so that was our hypothesis, that we're going to get a lot better immune response with this replicating RNA vaccine than you would with the one that doesn't replicate.
What's the natural half-life of RNA in the body, and do you package it to extend that?
Yeah, that's great. Thanks for leading me right into this slide right here, which is called this Lipid In Organic Nanoparticle. And RNA is very unstable by itself. And so it really does need to be formulated for two reasons. One, to enable it to get inside the cell, and the other is to stabilize it. Otherwise, it would just fall apart really quickly. And so we ended up working with this company called HDT that developed this Lipid In Organic Nanoparticle, or what they call LION, to help to deliver this replicating RNA. And what it is just basically this sort of a lipid sort of particle that gets produced. It can be manufactured at very high quantities. I'm not going to get into all the chemistry of it, but the long and short of it is that when you mix it with our replicating RNA, the RNA attaches to the top of it. And this particular particle stabilizes the RNA so it doesn't degrade, but this particle also acts as an adjuvant. We talked about that it actually helps the vaccine become more potent. For this particular type of formulation, it can be easily scaled up. You scale up the RNA, very high doses, large number of doses, and you scale up the formulation, and then you have two separate vials, and right before you actually use it, you mix it, and they just dissociate. And so you get this simple, quick manufacture. You can make more doses much quicker. But it's really important for RNA vaccine. That was really the breakthrough for RNA vaccines, was the development of these sort of nanoparticles. I'm going to contrast that both Moderna as well as Pfizer and J&J also have a nanoparticle. It's called a lipid nanoparticle. But how does LION differ from this lipid nanoparticle? What happens with lipid nanoparticles, they have to get their RNA inside that particle, whereas you just saw with LION, the RNA sits on top of the surface of our particle. Now, their process is much more complex. To be able to insert that RNA inside the particle, it's actually a process, and it takes, it's much slower. And they're still, I think, trying to figure that out, because that could be an impediment to really rapid scale up. The other issue is that they have to use cholesterol in the formulation. And what's not well known is that actually cholesterol is actually in very limited supply. And so this company that we worked with decided to make their particle without cholesterol, because all those components can be easily sourced, which means they have no barriers to a large scale manufacture. And then finally, I think really important is that because we can store these as two separate pieces, that sits on the shelf for a long time without degrading for months. Whereas once you actually get the RNA inside this nanoparticle here, they have to, right now, I mean, and maybe they'll figure out how to adjust that, they actually need a freezer to distribute that. And that then makes this particular vaccine dependent on the cold chain and could limit the distribution. So we were trying to solve some of the issues that we saw as potential barriers for lipid nanoparticles by coming up with a different nanoparticle that address those sort of things.
What about stability in the body, though? Other RNA treatments for biotech companies have had problems of getting into cells because the RNA degrades within the body.
Yeah. So that's how these now you're getting to an area that I don't know, which is how do these actually work? So we do know that these particular nanoparticles, they associate with the cells and they help to dump the RNA into the cells. So their function is not just to stabilize it, but it's also aids in helping to deliver the RNA into the cell. Some believe that it's just fusing with the cell membrane. Others say, well, it gets inside, gets taken up by the cells, and then once inside the cell, it's able to get outside of there's certain components within the cell that kind of sequester it, and it has to get out of that to be able to transcribe the RNA. I think there's a lot of groups still trying to figure out how they work, and that's the truth of vaccines. Sometimes we make a vaccine and then we figure out later how they actually work. But we do know that both of these types of technologies are very, very efficient in not only stabilizing the RNA, but delivering it efficiently into the cell. So that's been really the major breakthrough for RNA and why we're hearing all about them now. Without those nanoparticles, we would not be talking about RNA vaccines.
Dr. Fuller, just a heads up on time.
Okay.
It's 11:44. If you're able to pull the presentation part done within 5 or 6 minutes, and then we'll have another 10 for questions. I know you have more slides to get through, but we do have a lot of good questions I think will be coming in.
All right. I will probably not pause for questions. We'll just go through the last. I think we're a little bit more on the halfway already. So, anyway, we published this paper, and basically in summary, our particular vaccine that I just told you about, this replicon RNA vaccine formulated lion, was really exciting for us. It induced protective levels of antibody in both monkeys as well as mice. After a single shot. And those antibodies, even today, we're still studying those animals, are still sustaining for over 4 months, and that's going to be important. It induced T-cell responses, and I'll talk about that in a minute, and induced strong responses in aged animals. And remember, that's really important, I think, to have a vaccine that's going to work well in the elderly. And so let's talk really quickly about those T-cell responses. So as we talked about, antibodies are going to block the infection, but if antibodies fail to block the infection, there's these killer T cells that can actually find an infected cell and eliminate it. And so it's like this backup plan in case antibodies fail. And I think that antibodies won't always be 100% effective. So vaccines that are able to induce both of these types of immune responses are going to probably, in the long run, be more effective than only antibody-inducing ones. And so the big question is, how long is it going to take to develop these vaccines? So, traditionally it's been, at least 5 years once the vaccine got into a clinical trial, and can take, 10, 20 years even before that. And so usually what happens is that you have these, let me just go to the next slide here. What traditionally happens is that you first start your clinical trial, your phase 1, which is about safety, to see what and find your effective dose. And once it's done, then you start your phase 2, and that's a large additional groups of people, say maybe including elderly, to see if it works in them. And then once that's done, you start your phase 3 trial, and that's going to be your efficacy trial, where you put vaccine in some people and placebo in other, and you see whether or not the vaccine protects the people that get it. What's happening is they've compressed this entire schedule so that all of these particular phases are now overlapping. And so let's have a look at our accelerated timeline for our lion replicating RNA vaccine. As you can see, we've actually overlapped the phase 1, the phase 2, and the phase 3, okay? And so we're accelerating that, and you can see that's going to be within 18 months to 2 years. It's just starting right here. We're right here at this phase, and you can see that within a year, we could have this vaccine and, commercialized. What also is happening is that you see we started the phase 1, and rather than waiting until the phase 3 to start the scale up, they're already scaling it up. So they're doing this massive scale up of the vaccine, storing it in the warehouse with a hope and the expectation that the phase 3 trials will be successful. If they're not, and this is every company is doing that, they will either license it as successful, or they've got a whole warehouse of junk and they're just busted, okay? So what's important to know is that all these phases of clinical trials go through a huge amount of safety testing, and none of those safety tests are being skipped. What they're doing is just overlapping them, and that's why we're kind of in a new revolution of vaccines, where we're going to see vaccines being developed faster than we've ever seen them developed before. And so I think I would just want to kind of wrap this up really and talk about some safety considerations, because you're going to hear about that. Of course, every clinical trial addresses adverse events, and these could be just things that you would be unexpected, or somebody just gets sick or something from the vaccine, and that's always important to address. But there's other safety considerations for SARS-CoV-2 vaccines, and one is called antibody dependent enhancement of infection, or ADE. And what that means is that let's say your vaccine doesn't neutralize the virus as effective. If it doesn't neutralize it, then the virus can still attach to the antibody induced by your vaccine. And now that antibody, instead of blocking the infection, helps that virus get inside the cell. And if it helps it get inside the cell, then that cell is going to produce more virus, and then you're going to get greater disease. There's another concern, and that's something called vaccine-associated enhanced respiratory disease. And that means you have to induce with your vaccine the right type of response. We have two types of different responses that vaccines can induce. One can be pro-inflammatory responses. And as you may know, that SARS-CoV-2 is a disease of inflammation. Once the virus is even gone from our body, we have persistent inflammation that actually manifests itself as pneumonia and eventually fibrosis as well. So you don't want these pro-inflammatory responses. You don't want your vaccine to induce those, otherwise you might increase disease. Instead, you want your vaccine to induce anti-inflammatory responses, and that's going to help to reduce the disease. And we call those type 1 immune responses. And so you'll hear that vaccines are being tested. They say, hey, we induce Th1 responses. That's what that's all about. They're trying to avoid those Th2 type responses. And so where are we now? We have a bunch of vaccines in phase 1, and we have one already has been approved by CanSino in China. They've licensed it for limited use in the military. That's an adenovirus-based vaccine. You can see there's mRNA vaccine, there's adenovirus vaccines, and inactivated vaccines already in clinical trials. And as I mentioned, our vaccine is just starting into phase 1 trials. And so I don't believe there's going to be one single silver bullet. Okay, I don't think one vaccine is going to be able to do it all. This is all the things it needs to do. I think some vaccines might be able to induce immunity very quickly, but they might not be, as effective across different demographics. And so you'll have other vaccines that are very effective to scale up, but they may not be as strong in terms of antibody and T-cell response. So I think it's going to be a combination of multiple vaccines to be able to cover the different demographics, to be able to distribute it worldwide and to induce the best immunity. And even then, I think we're going to have second and third generation vaccines that are going to be coming on that could do better than the first generations that we see. But they're all going to play an important role. And sort of likens this to my son here, who's an archer. It's like, it's not one single bullseye that won him that national competition. It was multiple arrows getting very close to that bullseye that helped. And so once we have vaccines, how are they going to be used to stop the pandemic, and who's going to get it first? That's always the big question that's going to come up, and we're certainly going to probably talk about that quite a bit. But as a scientist, I think of it in a very different way than maybe the way politicians think about it. You have to get a vaccine, effective vaccine in billions of people. That's going to be a major hurdle. But if we have increased testing concurrent with the development of effective vaccine, there's this concept called ring vaccination that could be employed. And that is basically, you go wherever you've got the most infections going on, and you start vaccinating people that are the closest contacts, and the people within those populations, you start to isolate that particular disease in that particular area, and that helps to have a much more dramatic impact in starting to shut down the infection. So it's something to kind of think about as we proceed forward with these vaccines getting licensed for use. So.
Yeah, what a terrific presentation. Thank you. So clear. So forgive me, you may have addressed this. So I write for Lisa Krieger. I write for the San Jose Mercury News. What influences the persistence of the antibodies? We hear that some of them might be short-lived and others long-lived.
Yeah.
What influences that? And then forgive me, my second question is, where's yours going to be manufactured? Is there a facility someplace that's already making this?
Yeah, so the first question is that, basically there's a lot of complex immune processes that help that vaccine to be much more persistent. But it's really those memory cells that help an antibody response to be more persistent, so that the more memory cells that you produce, the more likelihood that that antibody will persist, and that in the event of an exposure to the pathogen, you'll have a quick recall of those memory cells, and they'll be able to quickly produce antibody. So in answer to your second question is that all of the companies, just like the HDT, are actually manufacturing their vaccines in multiple places all over the world, because once they're ready, then they'll be able to distribute them much more quickly if they have a plant in Asia, if they have one in the United States and Europe, and so they're all negotiating to have it produced all over the world. Yeah.
Thank you.
Mm-hmm.
Okay, Juliet.
I am Juliet Beverly from brainfacts.org. Thank you for the presentation, Dr. Fuller. My question is regarding microglia and how that's looked at. There is some evidence that in the olfactory bulb that microglia has helped stop infection spreading to virus and spreading to the brain. So I was wondering if this is being looked at in the development. a vaccine?
Yeah, that's a little outside my area. I actually haven't studied that, so I wouldn't feel comfortable actually answering that question. But I'll have to look into that. Thanks for bringing that to my attention.
Hi, Emily Mullen with 10. I'm wondering if the side effects we're seeing in safety trials of the RNA vaccines right now expected of this type of vaccine, and are genetic vaccines any more or less likely to produce some of those adverse events you mentioned, like ADE?
Right. Yeah. So for the first question, RNA is actually the genetic material of pathogens in general, viruses, and they have built-in sort of host stimulating coding sequences. And so there was always known coming into the trial that RNA itself could stimulate certain reactions that might cause an adverse event. And so those phase 1s were trials were specifically designed to find a dosing range in which that didn't occur. And I think both of the companies that have gone through those trials were very successful in identifying where that is. So, and I'm trying to remember your second question.
That was whether genetic vaccines are any more or less likely to produce some of the adverse adverse events you mentioned, like ADE.
Yeah, I think they're actually less likely, and that's because genetic vaccines can be designed to exclude regions of the spike protein that might cause that ADE, whereas other types of vaccines, you can't do that.
Okay, Angeli.
Thank you, Angeli from Yahoo Finance. I've been meaning to look this up about the adenovirus. There's immunity to that, so someone who's previously been vaccinated would not be able to give any kind of response to one that's developed now, right? That's correct. And has RNA been studied enough to know if that's the same for them?
Right. So with the adenovirus, there's a recent clinical trial by Cancino that actually showed that people who had pre-existing antibody against adenovirus failed to respond or respond very poorly to the vaccine. So that was expected. With RNA and DNA, because it doesn't have any immunity against the genetic material itself, we've never seen that. For the 30 years we studied that and tried to induce that, we've never seen a problem with that.
Okay, Andrew.
Hi, Andrew Dunn from Business Insider. I was wondering, you mentioned the idea of a second or third generation of vaccines. Do you think these would target other areas of the virus beyond the spike protein? There's a lot of concentration there, a lot of modalities going on, but all targeting the spike protein. And are there other differentiators to keep in mind with sort of a second generation of vaccines?
Yeah.
What are you hearing on that?
Yeah, no, that's a great question. That's kind of what we're working on in our lab, is now looking at other sorts of proteins, including ones that are much more effective in inducing T-cell responses, which I mentioned might be really important. I think the second generation ones are going to be designed with T-cells that play an important role in helping to sustain the antibody. And so if you start to incorporate more antigens within your vaccine capable of inducing that, that might help to make the immunity last a lot longer. The other aspect is that T-cells can cross-recognize different viruses within the same family. And so we have this vision for a universal coronavirus vaccine that, that's maybe the third generation that we come out with is going to be one that you're just going to take one and you'll have lifetime immunity, not just against this coronavirus, but any future pandemic by a coronavirus.
Okay, Sony.
Hi, thank you so much. This has been really, really helpful. I noticed that you mentioned monoclonal antibodies, and this is, I think, the kind of million-dollar question of how durable these responses are going to be to a vaccine. But speaking with a really broad brush, it does seem like we've been tempering our expectations for how durable immunity might last for a vaccine in recent months as we've learned more about the virus. And I was just hoping you could give us kind of your thoughts on how long, based on the data that you're seeing, we can expect these first generation vaccines to offer immunity, and how that might compare to some of the monoclonal antibody approaches.
Right. So monoclonal antibody approaches. Oh, sorry, you froze up there. Okay. So the monoclonal antibodies are not expected to last very long, maybe weeks at the most. And they're really designed primarily to treat the infection already, if you're already infected. Whereas vaccines are designed to actually give to people who are not yet infected and protect them from becoming infected. So that's an important distinction. With regard to durability, how long is it going to last? We don't know for sure with any of these vaccines until we've actually tested them. But I can tell you that for vaccinologists, our goal is to do better than nature. So you're seeing in natural infection that the antibodies might be waning quickly, but vaccines are designed with their adjuvants and other things we talked about to try to actually do better than that. And so far, at least in our vaccine, we're seeing persistence for months now after a single immunization that is much longer than what's been seen for convalescent patients. So we're hopeful, but we won't know until we test it in people.